Thanks for the introduction, and thank you for accepting my abstract to present at this conference. It's been a minute since I left the continent, and I thought it would be a good time to come back and reconnect. So I'm going to take you through a different journey from the one we've just witnessed over the last two talks. I'm going to delve into more the geekery of bioinformatics. So if you bear with me. So this work is really to address the question of identifying information from complex data that are generated by analytical instruments. So this is how we frame it. Bioinformatics for functional genomics essentially requires that we identify information from a variable space that is inflated. So generally, what we have is a scenario like this. You have, so the expected value of y being your phenotype, given x, your measurements. So your measurements are populated by a multivariable or a multivariate space, which is usually gene expression, multiple genes, multiple proteins, multiple metabolites. And these data usually are acquired using analytical instruments, which we sometimes call bioanalytical instruments. What's the challenge? The challenge then is to identify information, essentially identify patterns from your high variable space that represent your phenotype. In other words, regress X on Y. And the general paradigm that we take is that of a linear relationship. In other words, we relate X and Y in a linear form using some kind of a model that maps X to Y. Any exponential family of distributions or parameters can be used. Now, of course, the goal is to get information, given hypotheses and data and so forth. So what's the problem here? The problem is that we have large data and we have minimal information. In other words, the curse of dimensionality. We have developed a few approaches to mitigate these effects or to get this information from such data. Some of them are listed here, and you can access them from our website. All right, so how do we go ahead? Did we go forward already? I mean, we did, yes. So, no, I haven't. There we go. So here's how the data are typically organized. Usually you have your genes and your samples, or I can transpose this matrix so that I have samples of the row-wise or column-wise regardless. And so for every gene that is measured, it's related to a sample. So the goal is to identify patterns that are that represent the classification of these samples, given some biological phenomena. Now, the ideal way is to do what is called usually a bilinear decomposition. A bilinear decomposition is simply a way where you take a matrix X and you decompose it into two lower dimensional matrices that have got equal rank to the original X matrix, so that you get rid of the noise. And the noise that accompanies this is assumed to follow some kind of a statistical distribution, usually referred to as independent, identically distributed with a normal or identically distributed and following a normal distribution centered around 0. Okay, so this is a bilinear decomposition. The easiest way is principal component, where T here represents your principal components and V is your loadings, that's your variable space. There's a caveat though. The caveat is that those errors, the measurement errors, are assumed to follow some kind of normal distribution. So in a simple representation, let's take it like this. So this is a bilinear decomposition, essentially, where I'm trying to get the function that maps X to Y with the assumption that these errors follow some kind of a normal distribution, so that if I can estimate X from my data, then this is the band of uncertainty that accompanies my estimate. Unfortunately, this is not usually the case because some of these measurements do not have that normal distribution, or the uncertainties are not normal. So we've developed an approach here called error model gene expression analysis, which you can download and use. And essentially, instead of projecting the measurements or the solution onto the trial model, we project it in the direction that maximizes the uncertainty. Okay, we can talk about this offline if you want. Now, one of the main challenges, again, is that in the context of omics data, measurement uncertainties dominate, and they disproportionately affect the low-intensity signals. The high-intensity signals, you've got no problem. And the problem then is that when people have known this, and so what we do, you say we are going to model the highly variable signals. What does that mean? That means that only the dominant signals are going to be used for downstream biological interpretations. The low-intensity signals in a biological context have got the exact same, if not more, information than the dominant signals, because the low-intensity signals are the ones that inform the biology, right? They inform what happens next. So our goal then is to see if we can model these low-intensity signals. So if you cut off and take the low-intensity signals, you get a garbage like that. All right, so what do we got here? So we then developed an approach called kurtosis-based projection pursuit analysis. Essentially, what it does is like this. Let's take this as a simple regression model, and we identify your first principal component. Your first principal component is basically going to be influenced by the high leverage points, the outliers, and so forth, when indeed your solution should be there. If you don't know that you've got outliers, especially at the low intensity signals, you're going to end up with the wrong model. So you have a systematic bias in your estimate because your model is influenced by high leverage points. What does kurtosis do? So this paper is actually under revision, and if there's some pesky reviewers here, please, okay, be nice to me. All right, so I've just we've submitted the second round of revisions. Now, what does kurtosis do? So kurtosis is, instead of chasing variance as your projection or as your interesting directions of projections, we look through the data to see what other interesting projections exist, and where those projections can then be used to determine the patterns in the data. Those projections can be influenced by any of the central moments of tendency: kurtosis, skewness, Gaussianity, and so forth. So in this case, we've picked kurtosis. How do we proceed with our model? We take omics data. Unfortunately, I can't get a pointer here, so I'm just going to wave around, okay? So the first thing we do is we take data, multi-omics data, and we go through this process where we select the variables, we develop a model, we identify classes, we reselect the models, and then go back and feed it through a random forest classification, because KPPA, kurtosis-based projection analysis, is a classification or an exploration method. We don't know what clusters we have. All we see is the data separated in 3 or 4 or 5 clusters, but we don't know what those clusters are. And it does not give us what the variables that are important in the separation are. So we used a couple of data here, or 3 data sets, to test our model. The first data is the so-called the Aurora data. These data are breast cancer data, primary versus metastatic breast cancer, TCGA data of breast cancer, and Takemon. We call this a Takemon data set because it's developed by a group, published by Takemon et al. And they consist of methylation, RNA sequencing, and proteomics. And we've used all this data. The descriptions for the data are accompanied here. Okay, so how does the workflow for KPPA run? First, you get the original data, you randomly select variables, you perform a dimensionality reduction, you determine the class separation using KPPA, determine what those classes are, because this is unsupervised. So we determine what those classes are, and then select the features that are important using random forest classification, record those features, put them back into the original matrix, and store the important features somewhere. You run this 1,000 times, essentially it's doing 1,000 experiments, 10,000 experiments, and then you vote. You decide which variables have been picked the most out of your iterations, and then you continue with your functional validations. So what's the problems? The problems are two. One is that high signals or high intensity signals dominate the methods for data analysis, yet information is also buried in the low intensity signals. So the first thing we did is we say, okay, let's cut off the high intensity signals and focus on the low intensity signals. So these guys over here. The second problem is effect size. So essentially, if you do an experiment where the effect sizes are so big, you don't really need to do math, because I can tell in here there's men and women, okay? Now, if I bring in a mouse, the difference between men and women becomes very small. The mouse is big, okay? That difference. So that's what's called effect size, okay? So in a context of a biological experiment, usually biologists are going to design the experiment so that the effect sizes are really huge. That experiment is not risky and doesn't have very high rewards. So you want an experiment that has got very small effect sizes and to be able to identify differences within those types of experiments. And it's a little difficult to generate such experiments, so we carried out a simulation for this one. Now, here's the result for our, this is the result for our low intensity signal. So here's this heat map. Essentially, this is a benchmarking exercise. We took the most common methods used for classification, and we identify how best they perform using what is called silhouette coefficient. And a silhouette coefficient is simply a metric that is used to evaluate the quality of clustering. All right. Now, if you can see here, this is KPPA. So the higher the silhouette coefficient, the better the method is, or the clusters are. So as you can see that KPPA is performing way better than everything else at variance levels of 15%. So 15% of the lowest variable signals, okay? At high variable signals, of course, it also performs well. There are some methods here that have got issues. Now, this is the classification. So this is the classical classification plots that you see. So this is data for the Takemon data. So PCA, the first two principal components, identify 2, 3 clusters maybe. But you can see from KPPA that you can see all the 4 clusters that accompany this data. And these are the other methods that also are available in the public domain. Then we did the effect sizes. So effect sizes, essentially what we did is we created this data and took out a few features and reduced their differences by 0.1, 0.2, 0.3, up to 5. And so, as you can see, as we go along, most methods cannot access those differences until you get an effect size of about 0.5, and you can see that KPPA is working quite a bit better than that. Now on this here, so then we asked KPPA to identify the signals that we have artificially changed. And this is called the true positive rate, essentially. And as you can see, KPPA can pick up those effect sizes or those features quite well. So for TCGA data, for breast cancer, we decided to check the classification of different breast cancer subtypes using what is called the PAM50 panel of genes. And as you can see, the original PAM50 panel of genes classifies the breast cancer subtypes like that. If we include KPPA, this is how KPPA clusters the breast cancer subtypes, actually working better than the PAM50 classification. Now, what do we got here? So this is additional breast cancer data. So that's the PCA type plot. And you can see up on C, PCA is not doing very well. You can see some clusters, but when you look at KPPA, it's a very clear, distinct clusters. Over here on the heat map, you see the genes that are separating the PAM50 clusters along with what the stars indicate the original PAM50 panel of genes. And finally, we then apply this to a different data set. This is the Aurora breast cancer data set, where we're looking for 2 classes, primary and metastatic breast cancer. And you could see that kind of classification. But then when we got down to look at actually everything else, we identified something quite unique in that the tumor mutation burden is actually the biggest cluster, or the biggest source of classification of the data, when we initially thought it was the primary versus metastatic component. In summary, here's what I have. Analytical errors have a significant effect on omics data, and variable or variance-based methods cannot model data, especially with outliers of unknown origin. The dominance of high-intensity signals obscure low-intensity measurements, although these have significant biological importance. KPPA-CAT can model large omics data with small effect sizes and reveal meaningful biological information. A caveat, though, you wouldn't run that on your laptop. Okay, so the minimum we've been able to run KPPA on is a computer of maybe 12 cores or so. Okay. Finally, I would like to acknowledge my research group. So this work was driven by Fabian Bong, who's just a master student that defended recently, and Jasmine Bara. Oh, there's a guy here from UCT. I think you must know Ibrahim Ahmed from UCT, and Nidhi, of course, is in India. And these are my founders. And thank you, and I will welcome some questions.
Thank you, Dr. Tobias. Again, we pressed for time, so I'll just allow one question.
Yeah, thank you for this really interesting talk. Can you give us some feeling about hyperparameter tuning, sensitivity to such kind of parameters?
Yes, yes, we do some parameter tuning, except that we have made it automated, so that within KPPA, we minimize some function that decides how many features you need, essentially. The problem is because we are putting it through random forest, and random forest is by definition stochastic. You are going to. That's why we do several iterations, right? So that you end up with just really the most stable features. A very good question.